Imaging studies reveal that many brain circuits that normally regulate mood are dysregulated in depression. Located deep within the brain, the amygdala processes highly salient stimuli such as rewards and potential threats. In depression, the amygdala is overactive and responds excessively to negative events. In turn, the amygdala connects to a set of brain regions that hone the physiological and behavioral response to emotional stimuli. These areas include the medial prefrontal cortex, the nucleus accumbens, the hippocampus, and the insula. The hippocampus is involved in memory formation, and along with the prefrontal cortex, is particularly vulnerable to the effects of stress. Depressed people are more susceptible to stress, which can cause physical changes in the brain, including atrophy of the hippocampus. This and other changes in depressed people may cause inappropriate responses to emotional events. The medial prefrontal cortex is involved in regulating how strongly we react to emotional stimuli. Treatments such as antidepressant drugs, cognitive behavioral therapy, and electroconvulsive therapy affect the structure and function of these and other brain regions. Animal models such as mice are critical in helping us understand the cellular and molecular changes underlying depression and develop better treatments. Although it's impossible to know if a mouse is depressed, mice subjected to chronic stress show some symptoms similar to depressed humans, such as anxiety-like behavior, less social interaction, and a lack of interest in normally pleasurable activities. While not all human depression is triggered by stress, these models may still shed light on the biology of depression, and they're the closest scientists can get while studying mice. As with humans, chronic stress in mice can lead to atrophy of the hippocampus and prefrontal cortex. Mouse studies have also shown altered neuronal plasticity in various brain regions, including the hippocampus, prefrontal cortex, amygdala, and nucleus accumbens. In a healthy hippocampus, experiences can lead to changes in the connections between neurons, resulting in learning. These changes are referred to as plasticity. Chronic stress can reduce this plasticity. Healthy brains also continue to produce new neurons in one part of the hippocampus. These new neurons slowly mature and integrate into the circuitry, where they have a strong effect on hippocampal activity and behavior. These new neurons are also affected by stress. They're reduced in number in stressed brains. These effects may result from reduced levels of neurotrophins, proteins that increase neuronal growth and plasticity. Reduced plasticity may stop the hippocampus from being able to properly regulate the stress response, which may lead to a vicious cycle where stress perpetuates more stress. The hippocampus is particularly affected, but there can be reductions in plasticity elsewhere in the brain, and together these changes could contribute to other symptoms of depression like anhedonia. Whether these cellular changes seen in mice are involved in human depression remains unclear.